All right. So just want to introduce our next lecturer, Dr. Madeleine Pahl, who's a nephrologist at the hospital, also very involved with the Institute for Clinical Translational Science. And she's going to talk about both the barriers to translational research, which you've had some exposures already through different lectures. And then subsequently, we're actually been talking about this for quite a while. We're going to go over some of the examples we've given you and do some informed consent simulation sessions for you and have you work on them individually among yourselves as well. So thank you, Dr. Pahl, for being here.
Thank you, Dr. Lotipour, for inviting me. And I thought I would start today with a little introduction to the barriers to translational research. And then often people consider those as sort of regulatory barriers, which is when you move into this sort of IRB world and where informed consent takes a role. And so today, the bulk of our time, I thought, would be spent doing some workshop type, interactive, educational process where you get to practice obtaining informed consent in a sort of a predetermined studies and sessions, and hopefully that'll get you feeling like, yes, I can do this if I ultimately can participate in some research in that sort of active process. But before that, I wanted to cover simply what people talk about, and it's become so popular. What are the real barriers? Why haven't there been more clinical translational research studies that result in cures? Why haven't we cured cancer? Why haven't we cured HIV? You know, the things that they talk about in television. Why can't you grow a kidney, please, and put one into me? Well, there's a bunch of issues that are involved in obtaining information from the bench to the bedside, as it be. And I thought we'd just cover this, and this kind of, many people have written about this, and I like this sort of a cute diagram that's been described by some investigators. And if some of you are biology students, you'll recognize the cell cycle. But in fact, it's used here to describe a translational research cycle. And if we could outline it first, then we can talk about what are barriers to this process. The first one is somebody has to have an idea. That's T0. If somebody doesn't think of something exciting, then there's not going to be any research. Once you get this bright idea, someone has to then design studies. Usually these are basic science in the lab studies where you try to find a discovery. You discover a pathway that's involved in something, the effects of one transcription factor on cell behavior in a disease or even in normal physiology. Then that discovery has to be translated into something that's appropriate for medical knowledge. So you find a pathway in a cell. Does it have anything to do with glucose transport, right? And then you would imagine, okay, if I find a pathway that has something to do with glucose transport, it may have something to do with medical problems. And then you would have to apply that idea, medical knowledge, you would have to apply that idea to a problem. So you would say, sure, glucose transport may have something to do with type 2 diabetes. And then you would be talking about a medical problem, there would be studies, etc. And ultimately, if you identified some pathway that may be blocked with some agent or enhanced in some manner, then you would move into clinical testing. And that's this kind of idea that this circle puts together. Okay? So now you don't need to have a lecture to start to imagine all of the processes that are required from going from one to the next, to the next, to the next, and ultimately terminating in a clinical trial. You could even invent some, but many have been talked about, and if you look at it in this manner, in this sort of of circle, you could imagine, obviously, you can't have an idea unless you have people who are educated on the problem, understand the process, and are clever, innovative, or maybe just darn lucky. But you need to have a group of people that are involved in the process. I don't know that we know very much about how to change that, and if you guys have some ideas, well, that's great news. I suppose the Greeks called it the muse, but in this case, we'll just skip right on to discovery. What are the barriers that block someone who has a terrific idea from then interrogating it through some sort of scientific process? You have to develop a hypothesis, right? And then you have to have access to the ability to test that hypothesis. And people talk about this creativity to design the appropriate study. This is more than just the idea, but the ability to design the appropriate study that tests the question, the access to the tools that allow you to test that, money, and collaborators that will facilitate. If you want to study a protein and you don't know how to do a protein synthesis, that help you with the design. So you could see those issues can sit in that area that goes from T0 to T1. Okay. Now, you have to start going, and many academic centers are very well prepared to do that, right? That's what every single person, faculty member at this university is doing in some way or other, in some manner or other, at least once in their career. Universities are very well poised to do that. But then, when you start identifying from T1 to the synthesis of medical knowledge, now you have to develop a relationship between the scientist, basic scientist, the biochemist, the chemistry professor, and those people who understand clinical problems. Somebody has to direct the question to a relevant process. And these are usually scientists that understand clinical medicine or clinical problems and can tell you that, but without having to spend 3 years looking at the literature, that this process that this person is looking at has a relevance because I understand how glucose is transported. And yes, this is important, and no, this is not a relevant process, and we don't need to focus on that. That requires a cultural change where there is interaction between one group of people and another. And that's hopefully what centers like the ICTS here at UC Irvine and other places do. They bring people from different areas together. Okay, that's one of the barriers people talk about there. Of course, again, all of the things we talked about before are also there. You have to be able to know how to design the study, you have to be able to have money to do it, you have to have the tools, etc. Once you identify those processes with these clinical scientists, you have to then to go from synthesis to application to the medical problem. Now you start to even step up that need to bridge that cultural difference between basic science and clinical scientists. In that setting, some of the barriers include access to human tissue, adequate animal models. Is there an animal model for your disease so that you can test these things? Do you have the right team of individuals now that need to help you to progress through this process? And now you start to bring in people like pathologists, internists, surgeons, okay, pharmacologists, biostatisticians, in addition to the other team that was the basic scientists in chemistry possibly, and a physiology professor at the medical school, okay? And then once you get through this, you have to start, here is, people talk about the valley of death in drug discovery. You have to, once you get past this, you've identified a pathway, you've identified an inhibitor of that pathway, you've identified that that pathway is important in the onset of diabetes, you have a drug, But now you have to start the process before what are called preclinical trials and finally clinical trials. And there are many barriers there, including the ones we talked about. The most important here, again, are the ability to test the agents, the ideas you have, the pathways you have in the right models, animal models. Doesn't sound like a big deal, but believe me, it is. In the right tissues, that you have access to human tissues, and then ultimately here, once you form these large teams, that you can get adequate funding. Now you're talking in the millions, 50, 100 million, 40 millions of dollars to 100 millions of dollars to bring these agents to clinical testing. When people talk about barriers to clinical translational research, they often focus on the regulatory issues that are here. And those are the ones that immediately come to mind. That's why I wanted to spend a few minutes telling you a little bit about the others, because the other ones are really much more important. The regulatory issues are really relatively easily, I shouldn't say that, but addressed if you approach the processes in the right manner. So if you have to get an IRB approval to do a clinical study, if you've done your homework before, you just need to do the application in a proper manner. If you want to get FDA approval to test a drug that has not been tested before or been approved, you have to have done this sort of previous cell cycle work in a manner that identifies need, identifies efficacy and safety in a small but important manner, so that you can go on to do this study. And so IRB, FDA, intellectual property issues, issues with health insurance, Portability and Accountability Act, et cetera. I started to write a variety of regulatory issues that people think about can be addressed. I think the more difficult is to get there. But today, our focus was to try to address in a practical manner some of these regulatory issues that people often talk about. So if you've had a product, and one of those that I put as a sample study, if you've had a product that you want to take to Clinc phase 3 clinical trials, you apply for appropriate INDs from the FDA, and then you go to do the study, and you have an informed consent. We want to discover the process that you need to do when you're out in the field obtaining correct informed consent so human subjects are protected. And so that is what I thought we would cover today in the remaining part of the hour. But I'd like to open up for any questions, comments, and yeah.
And one thing, so after the questions, that would be good, Dr. Pahl, if you want, before we get into this,
yes,
is if you could talk a little bit about your own field and your research. That might be of interest to them. And then if you wanted to maybe just give a little background on the 3 different samples that we have, and then
got it, right. So yeah, because I think what we're going to do today is going to take longer. I didn't want to take up all my time giving a lecture, so I purposely didn't make a bunch of slides or tell you anything. But I think some people wanted to have a little overview of this, so that's why I took the time. I'm a nephrologist. So that means I trained in internal medicine after medical school and subspecialized in nephrology, which is kidneys. We do not do surgery. This is an internal medicine specialty. So we're interested. Nephrologists are usually physiologists, care for patients with kidney disease, manage individuals with dialysis, manage kidney transplantation. So there's a huge area of interest in a variety of disorders in nephrology. One of the common causes of renal disease, or what we call end-stage renal disease, people who need dialysis in this country, is diabetes. And so you can imagine that if you have over close to 600,000 dialysis patients in the U.S. today, and half of them are diabetics, nephrologists become very interested in this problem. And one of the large more clinical trials that I've participated in, and it's actually been a 10-year endeavor, and we continue to find data, is a study looking at the genes or that genetic influence in the process or the development of kidney disease. We initially started looking at diabetes, and then in some non-diabetic kidney diseases, diabetic nephropathy, that's the name for kidney disease caused by diabetes, seems to be much more common in certain ethnic groups. And our team was part of a large consortium, an NIH-funded study called, with the acronym of FIND, Family Investigation of Nephropathy and Diabetes, that looked for some of these genes. And as I said, it's been rolled over 8,000 individuals. There's been several genome-wide association studies. Because over 10 years, you can imagine, the ability to do these studies has changed. That technique, that technological areas exponentially changed in that period. And there's been, interestingly, genetic association in non-diabetics of African American descent who have a very high propensity for kidney disease, for a gene now that explains 70 to 80% of the cause of renal failure in that population, which is an ApoL1 gene that sits on chromosome 22. So that study is one of the things that I'm interested in. We've stopped enrolling patients. We're now only analyzing data, but that's one I thought I would share with you. And yeah?
And just on the barriers, could you talk about maybe some of yours that would kind of put that in perspective
Right.
for everybody, some of the ones you've seen and overcome?
And this is one of these studies where the bright idea, I think, has been around for decades because we knew from observation, all clinicians, that people with certain ethnic groups get more kidney failure than others. So that if you're African American, your propensity for nephropathy is almost fourfold compared to European Americans. And if you're Mexican American, it's certainly 2 to 3-fold increase. We also knew that these populations are admixed. Mexican Americans, we know, are almost 60-40 European ancestry and Amerindian ancestry. African Americans are about 70-30 European ancestry and African ancestry, and there happens to be markers that are ethnic associated that can be used to sort out the genome. So I can tell you that if you are African American, that you inherited the little portion of chromosome 22 from your European ancestors or your African ancestors. So the bright idea was around. The discovery came because we were able to identify these different markers. I think those markers were around before anybody really knew they could be applied in this sort of manner. But at the NIH, individuals who understood genetics, diabetes, and the nephropathy decided to come together to write a request for proposals so that communities, academic communities in the United States could apply for grants to generate a study. This began in 1998. And so we were able to get right to the S part relatively easily because the NIH provided incentive for different groups to come together. Geneticists, nephrologists, endocrinologists, biostatisticians, and a bunch of mathematicians who handle this data, computer guys, we don't even know what the heck their names are, that handle trillions of data points, okay? So that got us to S right away because of a push from above. Otherwise, people wouldn't have done this. Now, proposals came from all over the United States. 8 centers were chosen. Everybody had a different idea how to get this together, how to address the problem. So now we have to apply the situation here. And let me tell you, that was a significant barrier that led to monthly meetings for 6 months before the consortium could decide on a design of the study that would encompass the tone of the RFA, so the request for proposals generated by the NIH. And ultimately, proposal was generated that would address a central question. But again, like everything in the world, no one would give up a little bit of their ideas. And so additional monies were apportioned so that individual centers could provide some additional research based on their proposals, okay? And then, now in this case, there's clinical testing, the equivalent of clinical testing. As data was identified, clinical testing in genetics, we're not making a drug that's going to block that gene. Clinical testing, at least in the short-term goal here, would be you have to replicate your data in another population. And that has been a difficult thing because you can imagine they collected thousands of patients, 8,000 in this. We identified genes that have certain areas in the genome that have certain association with the disorder. And now we have to test it in another population. So we have to make friends with geneticists, clinicians that have collected large number of patients in Europe, in type 1 diabetes out of Joslin Clinc, in other studies looking at, say, Mexican Americans' cardiovascular disease and diabetes, so that these data can be replicated. And at current, some of these data have been replicated and identified the abnormality in that genome in chromosome 22 that I told you codes for a gene called APOL1 that's associated with nephropathy in African Americans. And we're still waiting to do replication testing on Mexican Americans, and a paper will be coming out soon with genetics of that disorder, Mexican Americans and diabetic nephropathy. But that's a little example of a large study that involved pretty much that whole circle. 10 years. I don't even want to know how much money the NIH has put in. It's over 40 million. But I didn't want to say. Yes?
So one of the issues that we talked about is the research you described, even though it involves a large human subject pool, it's looking for genes, and we were trying to name different stages of translational science. So the ultimate goal will be either to diagnose early or predict or protect.
A good question.
So, yeah.
Right.
In this case, it looks very
Right.
basic science type. But
It does, very basic science type. So what has happened in the last 5 years that may be translated to clinical practice, although again, it remains to go there. For example, we now know in small numbers that African American kidney donors that have the APOL1 genotype that is associated with increased risk for nephropathy. If they're homozygous for that, there's a G2 and 3, doesn't matter, with the right SNPs, the outcome of that kidney transplant is less than those who do not have that genotype. In individuals, I think, so that would be something that would immediately change practice if it were embraced by the transplant community who never wants to give up an organ. In terms of diagnosing, I didn't bring that up because you understand the complexity and the social issues around identifying genetic abnormalities that predispose to a disease. And certainly in I would say a population that may be vulnerable because of previous issues, you don't want to identify African American individuals who may have risk for nephropathy so somebody won't insure them. But it certainly, the finding of that gene has certainly identified the individuals who will be at risk. You could turn the coin and say those that don't have the APOL1 SNP variations that are associated with disease are protected. And those could be considered if you were diabetic, family diabetes and hypertension, and didn't have that gene, you might not be at high risk for nephropathy. But there's still no change in the horizon to transform that into a therapeutic intervention. Yeah, we're sorry. Yeah. Okay. So the 3 studies that I chose to try to cover how you would obtain informed consent in doing things, I picked 3 types of studies. I have them right here. Okay. One type of study is a relatively low-risk study. This study, entitled Effect of Community Health Promoters on the Use of Gardasil Vaccine. This is a vaccine that's been FDA approved, that's been tested and recommended for young women and now young men to receive this vaccine in order to prevent human papillomavirus infection that can be obtained through sexual activity and that predisposes in women to squamous cell cervical carcinomas, and in young men, either they transmit the disease to non-infected women when they're sexually active, but they too can get squamous cell types of carcinomas in perirectally and penile. So it is a disease that could be significantly diminished with the use of the vaccine. So this study talks about whether, and people, as you know, have been resistant. Families don't want their children vaccinated. They don't want to look as if they're proposing that immorality and some sort of promiscuous sexual activity occur because now people are vaccinated. So there's been a lot of resistance, who would have thought, among families to have their kids vaccinated. And one of the ideas is whether in certain communities where these health promoters have been helpful, they could encourage individuals, and in this case when I wrote this it was women because it hadn't been approved for men, that they would come to get the vaccine. So this is a study that is relatively low risk, and then the consent would involve a variety of issues that we could talk about in a little bit when we learn what. The second study is a study that is a drug that is already FDA approved. And the title is Evaluation of Cinacalcet Therapy to Lower Cardiovascular Events. And this is actually a real study. This is the acronym of this study is the EVOLVE study. This is the study of an FDA-approved drug that's used in dialysis patients. Dialysis patients who have a horrible coronary artery disease risk that is even tenfold their age-matched control. So you talked about the heart failure and cardiovascular risk. If you're a 20-year-old dialysis patient, your risk approaches that of a 60, 70-year-old hyperlipidemic man. So it's an important issue in the dialysis community. And this FDA-approved drug, this study was designed to look at whether this drug, cinacalcet, which reduces calcium, phosphorus burden, and hyperparathyroidism, one of the causes thought to be one of the reasons that dialysis patients have such a very high risk of cardiac disease, that this drug could change cardiovascular outcomes in that population. Cardiovascular disease is a complicated thing in the dialysis population, so this is a simplistic overview. But I brought this study here because it poses an interesting question. What do you do if, to do this study, you have to do a double-blind randomized control study. But what if the patients that are entering this study who have the disease, which is the hyperparathyroidism, for which the drug was designed and approved, what if they want to take the drug outside of the study? And so that's part of the important consent here, when you obtain this consent, you have to address the issues that become inherent in such a drug. The next study, and I chose nephrology studies because that's what I know, is a drug that actually fits this beautiful translational research cycle process, where the drug called bardoxolone was identified actually sort of serendipitously in cancer research. It was MD Anderson-owned drug. They were using it in cancer. They identified the pathway by which this drug works. It is an agonist of a transcription factor called NRF2 that is a anti-inflammatory transcription factor. So when things happen in the cell that make the classic transcription factor NF-kappa B go to the nucleus and make the cell make a bunch of what are pro-inflammatory cytokines and generate reactive oxygen species, this drug blocked it. You know, you've seen all those commercials. This is like the acai berry or the stupid little blueberry drinks in the pharmacologic world. So because inflammation, clinicians knew that inflammation is a big part of diabetes, that it plays a major role in the progression of diabetic nephropathy. The study company was formed, bought the rights from the cancer people, and fast-tracked that baby through the application of a medical problem. There are ample animal models of diabetic nephropathy, okay, and brought it to clinical trials. The clinical trials started a year and a half ago, phase 3 clinical trials. The phase 1 and 2 had been completed, and in fact, I told you in the little study that the phase 2 outcome was so exciting among the nephrology community, and even though it had only a few, I think total number of patients tested on that, said it was 100 or something, was published in the New England Journal of Medicine. Very high impact journal, very difficult to get cases into. So the company launched the phase 3 clinical trials, which I can tell you just 10 days ago were closed by the Data Safety Monitoring Board, because after they reviewed the unblinded data, there were too many deaths and congestive heart failure in the treatment group, and they closed the study. So I just came from the meeting 7 days ago where the big bombshell was dropped. It looked like a funeral, and everyone is anxious as to how to come back to all these patients. There have now been over 1,800 patients enrolled in that phase 3 clinical trial. So now you have to come and tell them, oh, give us back the drug, and oh, by the way, if you're on the drug, there was more mortality. You know, people like to hear that. So I thought it would be very important to review how you obtain consent for a drug for which there's no real clinical experience. A couple hundred people just doesn't cut it. And how you make sure that people understand what is it that they're getting into, even in how we present that information to them and temper our own enthusiasm for what we thought was going to be a terrific, new, innovative treatment. So I know you guys already got the lecture on informed consent and have the whole plan on how to do it, and know what are the issues, and you've got videos and who knows how many things. But basically, right, your jobs, when you get informed consent, and you see you can't skip any of these things, otherwise this is what happens, you're stuck with mud in your face, is to make sure you get informed consent from a person who understands. You have to get a subject that's willing and able to give informed consent.
So
right?
one thing that might be really good as part of what you're talking about, if it's okay, Dr. Pahl, to go over the
yeah.
research consent case.
Oh, right. No, I was going to say, they were going to do it. I was just going to tell them this. Remember that? You have to give people, and if they're impaired, none of these are impaired people, so we don't have to cover it. 2, opportunity to give consent. You got to give people time. 3, you can't coerce them. Make sure you don't do that. You've got to make sure you give people time to answer questions, right? And make sure that people know that this is voluntary, what the purpose is. And then the rest of it is the stuff you already know about, right? How you explain the study, what they're going to do, who they can call, and all this rigmarole. So, what I thought we would do, and what we had planned in the past to do this, is to have people practice, okay? There's 3 studies, and there's 3 roles. It's just almost like everything's in threes, isn't it? The Trinity, anyway. Anyway, you don't want to go there. So, what are the roles? There's going to be an investigator who's getting consent. There's going to be a subject who's going to be the person obtaining the consent. And there has to be someone watching you to make sure, I mean, in this situation, because you're learning, that you covered all of the things that you were supposed to cover. And what we did, and that's what Dr. Lotipour is saying, ta-da, here's your little form. I'm sure I missed stuff that you guys can talk about among yourselves. This isn't supposed to be all-inclusive for everything. But you see in the front, there's the research consent case. Did the person getting the consent do these things, right? Did you tell them who you were? What's the role in the study? You can read it, right? Provide overview, describes the procedures, etc. This is the specific things a person has to do. And if you're the one that's watching them, you have to make sure they did it. And in the back, also, in this situation, if you're the subject at the end, we could give you a little piece of paper and say to you, did your investigator ask these things of you, right? And you could see that you appeared professional, gathered information, listened actively, etc. This, actually, this is, because standardized patients are commonly used to test medical students, find out if they're doing a proper exam, differential diagnosis. In fact, that's what this building basically was made to do. And what they do is they hire actors to play the role of the patient, and then the little lovely medical student goes into the room, and it's like a test. There's that patient sitting with abdominal pain, and you're being tested to make sure you don't miss the appendicitis. And in this case, we've transposed that technique for research and obtaining consent.